Okay, everyone. Can you hear me okay? Just give me a thumbs up if you're good. So welcome to my guest lecture. Thank you so much for inviting me to McCall MacBain postdoc series. Welcome to my lecture hall. This is in the VSB Lecture Hall 1, which is the virtual science building, which is a little bit like platform 9 3/4 in Harry Potter. It's right next to BSB, Basic Science Building, but this is VSB. And I'm going to be talking to you about Bayesian inference for this guest lecture. And so I just want to magnify myself a little bit here. And what you're seeing here is the logo for the sort of banner for my course on Bayesian inference, which I happen to be teaching this semester as well. And what's shown here is, I think, a really nice depiction of Bayesian inference, because those were patterns that I noticed in my driveway just a few weeks ago. The driveway was covered with a dusting of snow, and those look suspiciously like some sort of animal tracks. And I suspect that it's a rabbit. You can sort of see where the hind paws would be and the forepaws and the rabbit jumping down my driveway. And that's really what Bayesian inference is all about. It's about looking at data that we can observe, and then trying to hypothesize about what caused the data. So we look at data like that and we say, that's really curious. What caused that? And of course, there are a lot of possibilities, but if we know something about rabbits, we might say, well, there's a high probability if it was a rabbit that it could produce data like that. We're going to see in a second, those are called likelihoods. And then we also can work in probabilities like, how many rabbits are in my neighborhood? Well, there are actually a lot of rabbits. I see them all the time. So that's what we call a prior. So combining those two, we'd have an expectation for rabbits. And sure enough, that looks like it could be from a rabbit. So maybe that was a rabbit. So let me just get going with a little more detail about this. So I have different transitions I can sort of blow up my screen to. This is my larger screen where you can see me in the podium. And I'm just going to get started now in the full lecture screen. And I see we may have a little bit of a green screen issue here, which is one of the joys about doing this in our virtual lecture hall, is that it's virtual. So I think I just helped out there. Okay, that looks better. So let's continue on now with this introductory lecture into Bayesian inference. So what I'm going to do today is I'm going to talk about three different topics. One is just inference under uncertainty. What's the whole idea of Bayesian inference? It's we're trying to make inferences when we're not sure of the causes of the data that we can observe. And then we're going to talk about perception as Bayesian inference in particular, and then tactile perception even more specifically, which is what my lab studies as a Bayesian process. So first of all, inference under uncertainty. This was a problem that was of interest to Thomas Bayes, who was a amateur mathematician as well as a minister in the 1700s in England. And it was subsequently something that really interested the great scientist Pierre-Simon Laplace, who about 50 years after Bayes' death independently rediscovered what we now call Bayes' formula. And Laplace is really the person who put it into its modern framework. So Bayes and Laplace were interested in problems like the one depicted here. So this is a problem where you might imagine that you're in a county fair and there's a barn, and you're told that in the barn are 100 barrels filled with 4 apples each. And 25 of the barrels have 1 green apple, 25 have 2 green, 25 3 green, and 25 all green apples. And your job in this county fair is sort of game is just to be blindfolded, go in blindfolded, and until you just happen to stumble across a particular barrel. And then once you are there, you pull out an apple at random from that barrel. And then you back away, take off your blindfold, look at the color of the apple that you sampled, and try to figure out which type of barrel it came from. So we can call these different types of barrels hypotheses, because that's what we're really interested in this game. So I'm just labeling them H1 through H4 based on the number of green apples that might be in the barrel that you come across. So there are different probabilities that we can specify or talk about with respect to this problem, and some of them are very easy, and then the one that we really want to know is much harder. So to go to the easier ones first, before selecting an apple at all, when you just stumble across a particular barrel blindfolded, what's the probability that, say, it's this type, the H3, the 3 green type of barrel? So this is before you even take out any apple and look at it. If anyone would like to answer that question, feel free to raise your hand, and I'll see it in the participants list. Raise your hand in Zoom, that is, your Zoom hand. And then I can unmute you, and you can offer a public opinion. So what's the probability that the barrel you're going to come across is the H3 type? Before you even, just knowing everything you know about the problem so far, there are 100 barrels, 25 of each type. Okay. Bradley.
It would be 1 in 4.
Exactly. 25%, exactly. 1 in 4. And the reason is that there are, in fact, 25 out of 100 of that type. So you have a 25 out of 100 or a 1 in 4 chance of just happening upon that type. They're all mixed up randomly in the barn and so on. So that's a fairly straightforward probability to specify. It's called the prior probability. And we write it as P of H3, prior probability. It's called prior because it's before we even take into consideration the data, which is going to be the apple that we pull out, the color of that apple. So that's fairly straightforward. Now here's another probability. Let's suppose that we do pull out an apple and it happens to be green. So we back away, we take off our blindfold, we notice it's a green apple. Here's a question. If you were at this type 3 barrel, what would be the probability of drawing a green apple? So we write that as we state that as the probability of green given or assuming H3 is true. So who would like to offer me an opinion about that one? It's also fairly straightforward. It's not a trick question. Don't be shy. Right? So in H3 there are 4 apples and 3 of them are green. So what's the probability that I would pull out a green one? I'm very patient. I can wait here all day. I'm not going to move until someone gives me an answer. Okay. Leanne.
Mike put it in the chat, but I did agree with him. 0.75 or 3 and 4.
Great. Yes, exactly. Thank you. So 3 and 4. When I'm lecturing, I don't usually monitor the chat. You guys are very welcome to chat, though. Okay, so that's correct. So we write this P of green, and then this bar, which means conditioned upon or given that, meaning assuming that H3 is where we are, that H3 is the true hypothesis. So P of green given H3 is 3 out of 4. We call that the likelihood of the hypothesis. That's the probability, if the hypothesis were true, that it would give rise to the data we observed. That's a probability, for example, if that was caused by a rabbit, those tracks were caused by a rabbit, what's the probability that they would cause tracks that looked exactly like what I saw in my driveway, right? So that's called the likelihood. Now, those two were easy to specify often in problems. But what Bayes and Laplace were interested in was something a lot more complicated. They were interested in, now that we know everything we know, what's the probability that H3 or some other hypothesis is actually the right hypothesis? And so that's called a posterior probability because it's after accounting for the data, after taking it into consideration. So this is written P of the hypothesis, for example, H3, given the data, given green, the data we observed. So that posterior probability, if you just think about it, the way you can think about a prior or a likelihood, then you probably won't be able to come up with a number. You can't just pull that number out of your brain the way you can for the priors and likelihoods. So somehow we have to calculate that number. And it turns out we calculate it from the priors and likelihoods. But we can't just arrive at that all by itself. It's not obvious to us. So to arrive at the answer, what is P of H3 given green, or P of any hypothesis given the data? We follow some simple steps to do a Bayesian inference problem. First, we list our hypotheses. So here I'm just writing again what the hypotheses are, H1 through 4. We list out what the data were, a green apple, and then we specify the prior probabilities for every hypothesis. So the prior probabilities are 1/4 for every hypothesis, since there were 25 of each type in the barn. Sometimes we call that a uniform prior, because it's the same for every hypothesis. Then we write out the likelihoods for every hypothesis. So as you can see, that's just the probability of getting a green apple if that hypothesis is true. So it's 1/4, 2/4, 3/4, or 4/4. And then what we do is we multiply each hypothesis's prior by its likelihood. And then we divide each of those products by the sum of all of them. So this denominator looks kind of complicated, but it's just that big sigma just means sum of the prior for hypothesis I times the likelihood for hypothesis I. So summed over all of them. So for example, if you multiply 1/4 times 1/4, add 1/4 times 1/2, 1/4 times 3/4, and then add 1/4 times 1, it works out to that number, 0.625. So we divide each of those products by 0.625. And that gives us the posterior probability for that hypothesis. And that would be, these are the posteriors that you would get. Notice that these posteriors sum to 1. There's a 1/10 posterior for H1, a 2/10 for H2, 3/10 for H3, and 4/10 for H4. 1 plus 2 plus 3 plus 4 equals 10. So these sum to 1, letting us know that, sure enough, one of those must be true. One or the other must be true. They sum to 100%. Just like the priors sum to 100%. But what we've done is we've learned from the data. We've done what's called Bayesian updating. We've started with a uniform prior, no opinion, equal priors. But now we have a pretty clear opinion that it's more probable that we're at H4 than at H1, for example. Because the denominator is just a constant, and it just plays a role of making sure that the posteriors sum to 1, it's kind of less important. And we will sometimes write Bayes' formula in a simpler version, which is the prior for a hypothesis times the likelihood for the hypothesis is proportional to, that's what this is, this sign, to the posterior for the hypothesis. So we leave out the denominator, we can no longer put equals, but it's getting across the idea that the hypothesis with a higher posterior is going to have a higher product of its prior and its likelihood. So we can write these out as distributions. This is just a bar graph of all the priors, and you can see it's flat or uniform, a bar graph of all the likelihoods, and a bar graph of all the posteriors. And let's look at those distributions a little more carefully. So we call this distribution of the prior probabilities of each hypothesis the prior PDF, which just stands for probability distribution function. We call the plot of all the likelihoods the likelihood function. So you can see this is the 1/4, 2/4, 3/4, 4/4, the likelihoods. Probability of getting the data if that hypothesis were true. And then these are the posterior probability distribution function here, posterior probability of every hypothesis. Okay. Now, we can look at plots like this and figure out which one is greater for the likelihood and which one is greater for the posterior. The hypothesis that has the greatest likelihood, so the hypothesis that, if true, would be most probable to give rise to the data we observed, is called the maximum likelihood estimate, or MLE. The hypothesis that has the greatest posterior, which in the end is the one that we're going to think is most probably in fact true, is called the maximum a posteriori estimate, or the MAP. So this may seem kind of boring right now because, as you can see, the MLE and the MAP are the same hypothesis, the one that has all 4 green. That was the most likely to give rise to a green. It had to give rise to a green if it was true. And we're also thinking because we got a green, probably that's where we were. It's a little more likely to have produced the data we produced, and we had no prior opinion in the beginning favoring one over the other. So that's also our MAP. But what I want to convince you of today is that, and illustrate to you, is that in daily life scenarios, we often have a different MAP from our MLE, which is an interesting scenario. And so I'm going to just change the game. Imagine going to another barn in the same county fair And in this barn, there are, again, 100 barrels of apples, but you're told that 70 of those 100 are of type 1, H1 barrels. And of the other 30, there are 10 of each type. So what I would like to ask you is, what is this going to change? Which one of these distributions is obviously going to change as a result of this new scenario? Who would like to provide me with an answer to that? Go ahead and raise your hand in Zoom. Mike. Right, exactly. So what's going to change is the prior, right? The prior is now, remember, it's the probability of encountering that hypothesis, that type of barrel, just when we walk into the barn blindfold and we stumble upon the first random barrel that we stumble upon. There's 70 of that type, so we're more likely to encounter one of that type. And because the prior is going to change, the likelihoods won't change, right? Whichever type it is, it has a certain probability of giving us a green apple, so that's not going to change. But because of the product of the two in Bayes' formula, the posterior will also change. And this would be what you would actually get if you went through the math. You would now see that because the prior so sharply, we call this a sharp prior, it sharply favors one hypothesis over the others. And because the likelihood function is not so sharp, it's kind of broad, what we call a broad likelihood, all of those barrels could give rise to a green. And none is really so much more likely to do so than the others. Because of that, when we multiply each hypothesis prior by its likelihood, the winner turns out to be this one, hypothesis 1. So in other words, even though we got a green apple, we think it's most likely that it came from the basket, the barrel with only one green apple in it. So we now have a situation where our maximum a posteriori estimate, the MAP, is the opposite of the MLE. And what I want to convince you of today is that this is how perception often functions. You can perceive something that's actually has not such a high likelihood, but it might have a very high prior, and that's going to influence your perception. So that brings us to the idea of sensory perception itself as a sort of unconscious form of Bayesian inference, where your brain may be trying to evaluate hypotheses, like what's going on in the real world, to create the perception that you have at this moment of what's happening. And scientists over quite a bit of time speculated about this, but the one who put it sort of most concisely was Hermann von Helmholtz, who was a great physicist, and also he was very interested in optics, but he was also interested in the physiology of visual perception. How is it that humans perceive the world through vision? And in his treatise on physiological optics, published in 1867, he said that previous experiences act in conjunction with present sensations to produce a perceptual image. In other words, he recognized that what we perceive is not just based on what's coming into our retina, our eyes at this moment, the present sensations, but also our prior experiences in life that might lead us to have expectations for what's most likely out there in the world. So in the modern Bayesian framework, we say this is the prior, the previous experiences inform the prior. The present sensations, that's the sensory data that go into the likelihoods. And then the perceptual image is the posterior. And often this is paraphrased as Helmholtz's idea that perception is a form of unconscious inference. So Helmholtz didn't actually use Bayes' formula or Bayesian inference, but he put forth this idea very nicely and concisely. A much less famous, really not famous at all scientist puts it in a sort of concise way that perception is the product of expectation and sensation. And I kind of like this because it's the product of, meaning it results from expectation as well as your current sensory input, sensation. But you see in Bayes' formula, it's also the product. It's a multiplication. So I kind of like that. Okay. So let's see some examples of this. This is a well-known illusion, Charlie Chaplin face mask illusion. And it's rotating around on a stick. This is a hollow mask. Right now you're seeing the hollow side rotating from your right to your left, but I bet that almost all of you, probably all of you, do not see it as hollow, and you do not see it as going left to right. You see it the other way. Right to left, you see it the other way. So what's going on here? Why is our visual system fooled by this? Well, the idea is that when we observe the world, our brains are constantly, in the visual areas of our brains, constantly and without our awareness doing something like Bayesian inference. So our brains are evaluating hypotheses for what it's seeing, right? So you could imagine two hypotheses. One is that this is a regular convex, meaning bulging outward face, and the other is that it's some bizarre concave face when you see the inside. The data, the sensation, the data is the image on the retina that is produced by this face mask. Now, I would wager that pretty much our brains have a very sharp prior in favor of bulging outward faces. In other words, P of H1 is much greater than P of H2 for us, because throughout our lives we see thousands and thousands, hundreds of thousands of faces, and they're always bulging out, except maybe on Halloween if you look at the inside of your mask or something. So we have extremely little experience with hollow faces. So we have an overwhelming prior bias, if you will, for a bulging out face. And the idea is that this prior knowledge gets encoded, the statistics of the visual world get encoded into our visual cortical areas through synaptic plasticity. And so we sort of burn in neural circuits that represent our environments. We've learned, and that helps us to, without even conscious awareness, to interpret the sensory image that's coming in to produce a perception. What about the likelihood? The likelihood is very broad, almost flat, because the retina is basically a two-dimensional sheet of photoreceptors. So it's taking the three-dimensional object from the world and collapsing across the third dimension to produce a 2D image, like a movie screen image. And a hollow face or a regular face can produce the same two-dimensional image on the retina, since you're collapsing across that depth dimension. And so the probability of getting the data on the retina that we got, given that it was convex, a bulging outward face, is about the same as given that it was concave. And now when you multiply the likelihood by the prior, because the likelihood is so much greater for convex, and the prior is so much greater for convex, and the likelihoods are about the same, the posterior, the map estimate, is that it's a regular convex face. So it appears that our visual system concludes for us unconsciously that that's through some sort of Bayesian type calculation that it's a convex face. If it's a convex face, I just want to play this one more time, because if it's a convex face that you're seeing, if the brain concludes that, it has to conclude that the sense of rotation reverses. Because look at what's happening now. The eye on the left, on your left, is rotating to your right. But the only way that can happen if it's a bulging outward face is if the whole face started rotating in the opposite direction. So it's as if the Bayesian brain is doing a hierarchical sort of conclusion here. First, it concludes it's convex, and then because it's convex, to make sense of what's happening, it has to change rotation direction. Okay, so this is just in a rather qualitative way how we might envision a sharp prior influencing perception in the visual system. Now, I'm going to be telling you about how we study and generate and test Bayesian models in my lab. I'm going to just be able to have time to give you a very brief taste of it. But I want to emphasize that it's very important to be careful when we design and conduct research studies, because as everyone knows, it's really easy to fool yourself when you do an experiment. So it's important for us to be really, really careful. And so what I'm going to tell you is what we did and how we're conducting Akasha's research study. Now, what I would first like to do is just ask you what I just said. So there are these blanks in that sentence, and I would like to ask someone, what did you hear me say? If you could raise your hand in Zoom, what did I say in those two blanks? Okay. Leanne.
Less confident about the first one. I thought you said we're doing what we're doing and how we're conducting Akasha's research study.
Right. That's great. Okay, so that's what I wanted you to hear me having said. And later you can roll the tape and verify that what I'm about to tell you is true. I spoke kind of fast without great articulation on purpose, because I wanted you to hear, I wanted to actually talk about auditory Bayesian perception. And so this was my excuse. And I wanted you to hear what we did. You got acoustic waves that vibrated your eardrums that get converted to action potentials in your inner ear and go up to your brain. Then your brain has to figure out what words are those. So those are hypotheses, right? So we think your brain is always doing Bayesian inference unconsciously for you in the auditory system as well. I didn't actually say what we did, but it's entirely understandable that you heard that because I gave you a prior. The context of the conversation was such that that's what you would perceive as the map estimate. In fact, I said what V did. Why would I say what V did? Because some of the experiments I'm going to talk to you about were done by V Nyo in my lab. And there she is, that's V. And also, I didn't tell you that we were going to do, or how we did a cautious research study. This is even trickier because I actually said how we did a cautious research study because Akash Deep is another student in the lab who was involved in these experiments. So I gave you a prior that obviously your prior expectation would have been much higher for we and cautious than V and Akash's. You had no way to have a prior really for these other ones. So it was very sneaky of me. But what I really want to get across is that auditory perception can also be modeled as a Bayesian inference process. So for example, if you think of the data as the acoustic input to your ear, and you think of the brain as evaluating many hypotheses, these are just two of very many hypotheses, of course. And the hypotheses are what was the sentence spoken, what were the words. Given the context of the conversation, like what you expected to be in the barn, right? There's a context to everything. So in real life sensory experiences, and you would have had a much higher prior for the word we than the word V. That would have changed if I'd already told you that I had Akash and V were in my lab, but I didn't mention that. So the likelihoods, the probability of hearing what you heard, getting the acoustic signal you got, if I said we, or if I said V, they're very similar, but actually I'm putting less than or about equal to here. In other words, since I really did say V, it's a little more probable to have produced the sound that you heard than if I'd said we. But those are such similar words that these are roughly equal. So you have a rather broad likelihood function and a sharp prior, just like in the face mask illusion. And when you do the multiplication of the prior, the product of the prior and likelihood, the one that wins out in the posterior is hypothesis one, what we did. So the graphs would look something like this. It's hard to know how to quantify these. This is just a qualitative argument at this point. But the prior is very sharp in favor of we. The likelihood function is close to equal, so rather broad for the two hypotheses. And then when you multiply the map estimate, the posterior, the hypothesis of the highest posterior probability is we. This would have changed if I had articulated, enunciated very carefully. If I had said, et cetera, I'm gonna tell you about what V did, right? Then you would have had such a sharp likelihood function because the data coming to your auditory system would have been so much more consistent with being produced by the word V than we, that would have overridden your prior bias for we when you did the multiplication. Basically, this difference is even greater than this difference. So you would have actually had a map estimate that was equal to V in this case rather than we. But that has to do with the quality of the sensory data. And what I would like to convince you of is that in many real-life perceptual scenarios, the sensory data is of quite poor quality. One example is on Zoom. I'm sure you've all heard people breaking up. Hopefully I haven't done that today, but because of connection issues, that can happen. But even if you're with someone back before COVID times when you might go out to eat in a restaurant with your friends, or you're walking on the street with your buddies and a bus drives by, there's all sorts of ambient noise. You hear a word that's sort of difficult to make out, but because of the context of the conversation, you have a high prior for certain words. So it's very, very common to see examples like this in auditory perception, where priors are important, and the prior in Bayesian inference has more influence when the data are not really super clear. In other words, when the likelihood functions are fairly broad, fairly equal, that's when you do the multiplication, the prior just naturally has more influence if you have a sharp prior. So there are entire Bayesian models of speech recognition And people who were investigators who were trying to model mathematically and simulate in a computer how the human brain can recognize speech. So one thing that's quite clear is that we humans are still better than even any computers at this, and we really know how to recognize speech. So I'm guessing that most of you just heard me say, and I can see Joe smiling because he has heard this many times before, probably most of you have heard me say we really know how to recognize speech. But that's also not what I said. I said we really know how to wreck a nice beach by littering all over this beautiful beach. So this is just yet another example. I provided you with a prior. I was talking about speech recognition, but I all of a sudden said something else. Okay. And the something else I said had a similar likelihood, right? But so you perceive the one that had the higher prior. All right, so with that very rapid introduction to Bayesian inference, I'm now going to talk to you a bit about what my lab does, which is we study tactile perception, the sense of touch. And we produce Bayesian models to try to understand how the brain might be perceiving through the skin, perceiving the world, the tactile world. And we test these models experimentally against non-Bayesian models. So I'm just going to give you a brief flavor, and it may get a little technical here, so don't worry if you don't understand everything. You're not going to be tested on this afterwards. Not a real class, so I have some license to get a little technical. So one thing that we study in my lab is illusions. So most people are aware there are a lot of visual illusions. There are also acoustic illusions, auditory illusions. Most people are less aware of the prevalence of tactile illusions, but there are many of those as well. And we study one type of illusion that we call perceptual length contraction. So this is a rather interesting illusion that if you take a part of the body, especially one with poor spatial acuity, where you don't have a lot of receptors, for example, your forearm. This won't work on the fingertip. It works well on the forearm, though, or the back, areas of the body with not very many sensory receptors. And what we're going to do is we're just going to tap twice on the skin, once here and once here. And so if we tap twice, and this is done with electromechanical stimulators under computer control, and those two taps were separated by about a second, and then we ask people using psychophysical methods, sort of quantitative methods, we figure out where did they feel those two taps. Then from the stimulus I just gave you, there's not going to be any illusion. They feel them where they were produced, on average. There's some variability anytime you tap someone. They might be plus or minus a centimeter from where you tapped and what they feel. There's some noise in the system. But on average, there's no illusion there. But what's really curious is if you decrease the time between the taps, so here are two taps separated much closer together in time, then the perceived spatial separation reduces. And if you reduce the time between the taps even further, the spatial separation that's perceived reduces even further. So there's a very strong influence of the time between taps on the perceived space between them. And this is really curious. And it gives rise to even more cool illusions, which I think are really it's the same underlying principle, but there's a famous illusion among tactile researchers called the cutaneous rabbit illusion. So here I am talking about rabbits again. Another reason I like those rabbit tracks in my driveway for this talk. But the cutaneous rabbit illusion, we're going to tap 4 times, but only in 2 places. We're going to tap twice here, and then we're going to tap twice here with our mechanical stimulator. But the time between the second tap here and the first tap here is really short. So they're going to cause that perceptual length contraction. And what it's going to feel like is a rabbit hopping up the person's arm. So here we go. Tap, 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 tap. And what's perceived is tap, 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 like a rabbit hopping up the arm. So this was actually discovered in the 1970s by 2 researchers, Goldreich and Sherrick in Princeton University. And they discovered it by accident. They were playing around with various types of stimuli on the arm, and they messed up how they programmed something into their stimulator and produced this sequence. And then they were startled and said, who let the rabbit out of the cage or whatever? And then they proceeded to study it for many more years. So it turns out when we looked back in the literature that going all the way back to the early 1900s, illusions of similar sorts had been described. And despite many studies, these are just a few of the ones shown, no one had come up with a unified explanation for what might be causing these illusions. So just to remind you, the fundamental illusion is that if you were to give two taps separated by a certain time, much less than 1 second, so time here is on the x-axis, and position is on the y, so these are the two taps in position. This is just some arbitrary scale in centimeters along the arm. You might imagine, for example, lower numbers are closer to your elbow and higher numbers are closer to the wrist. So this is just showing two taps, in this case separated by 10 centimeters and about 0.2 seconds on the arm. And we can talk about the time between the taps, 0.2 seconds in this case, and the distance, which we call L for length, which in this case is 10 centimeters. And then we can talk about what's perceived, which we call L star. L star is the perceived distance, the perceived length separating the two taps. So over the course of decades, there have been many studies on illusions like this. And when we reviewed the literature, we noticed two very prominent findings. One is that a number of researchers had kept the time between the two taps equal, but changed the actual distance between the two taps. So they manipulated L by spacing the stimuli further apart or closer together on the skin. And what they found is that the perceived distance, L star, was always less than the real distance when the time was short, but it increased linearly as they increased the real distance. So here the real distance was being increased from 0 to 15 centimeters, and the perceived distance increased as a linear function of the real distance, but it was always less than the real distance. So this identity line here is where the perceived and real would have been equal, and you can see the perceived was always less than that, always less than the real distance. So that's one observation, that there's that linear relationship between perceived and real distance at a fixed time between the taps. But another one was the effect of time. So some researchers kept the distance, the true distance, the same. In this case, 10 centimeters was a true distance for all of these data points. But they adjusted the time between the taps. And what they found is that as the time between the taps increased, the perceived distance L star approached the true distance asymptotically, so it converged to the true distance. But as the time between the taps decreased, the perceived distance L star plummeted in a very nonlinear way down towards 0 perceived distance. So basically there's this linear effect of the real distance on the perceived distance, but there's a very nonlinear effect of the time on the perceived distance. And we wondered, why is this so? So we got curious, and that became our research question. And so we set about to construct a Bayesian representation for what the brain might be doing, and this is a speculation. This is our attempt to model this process and see if it could be understood as a Bayesian process. So first, we understood quite clearly, it's very obvious that there's a wide likelihood function when you're tapped on the arm, because when you're tapped on the arm, because you have very few sensory receptors on the arm, and because the neural response is also stochastic, and I don't have time to get into this, but the firing rate of neurons in your somatosensory cortex and in other cortical sensory areas is somewhat random. There might be an average amount of action potentials, imagine an average amount of number of electrical impulses, but on any given trial, there's a lot of variability from how many you're actually going to get. So because of that, there's a lot of uncertainty that the brain just has in the information provided in terms of electrical impulses in the brain in response to a stimulus. So we can model this with model neurons. So these are neurons that have areas of the skin that they respond to. They're simulated cortical sensory neurons. And when you tap on the simulated skin of the model, there's a process that produces action potentials. And then the brain tries to figure out where was the tap that was most likely to produce the action potentials in all those 100 neurons, for example, that we're modeling. And so this is a likelihood plot, just like for the apples. But in the data now are action potentials in a population of neurons. And because we have two different possible tap positions to figure out, this is position 1, which was in fact at 2 centimeters in this example, and position 2, which in fact occurred at 4 centimeters in this example. But we're getting action potentials, feeding them into our model, And the model's figuring out, based on the action potentials I got, what's the most likely. So that would be the MLE, the center of this graph. And what are the least likely? That's the very dark areas. And what are the sort of intermediate ones? Those are the blue. That might have given rise to that group of action potentials. So this is the probability of the neural response given every possible tap 1 position, and the probability of the neural response given every tap 2 position. And we put those together, and then every pixel in this square, every little pixel, there are thousands of them, represents a hypothesis for tap 1 and tap 2, what just happened on my skin given the action potentials. So this crosshair is what really happened. We tapped at 2 centimeters for tap 1, then 4 for tap 2. But on any given trial, because there's a sort of stochastic, a little randomness in the neural activity, the likelihood bounces around because the sensory data is different. You're not always drawing a green apple. The pattern of action potentials is a little different each time. And so I'm just showing repeated trials here. But you can see that on average, the likelihoods are centered at the true tap location. So the maximum likelihood estimate is centered at what really happened on average. But on any given trial, it's going to bounce around randomly. And you can see that there's sort of, you can imagine this as sort of a blob of uncertainty. If you want to think of it, you can think about what neurons are responding the most in the cerebral cortex, and it's going to create sort of a blob. You're not really quite sure, but you're going to think that whatever you get the most action potentials, that's most likely where the stimulus was. That's going to be the maximum likelihood estimate. But you have a blob of uncertainty indicating that a lot of possible tap 1 and tap 2 positions could have given rise to the neural data that occurred from the arm. For right now, I just want to point out that we model this as a bell-shaped curve in both dimensions. So this is what we call a 2-dimensional Gaussian function, a 2-dimensional bell-shaped curve. And the standard deviation, which is essentially just the width of the curve, or the width of the blob, is called sigma sub s for spatial sigma, spatial uncertainty. So the bigger that blob, the more uncertain the likelihoods are, the more different positions on the skin could have given rise to that same data that the brain got on that tap. So we then thought, well, that's the likelihood part. That's actually fairly straightforward to model with our computer modeling. But what about the prior? And here it becomes much more speculative. But what might people expect to happen on their skin? Well, we don't know. But if you just sort of think about it, we don't have data. We haven't tracked what touches people's skin on their arm throughout their lives, for example. But just thinking about it, most of what touches us is either stationary or very slowly moving. So for example, my shirt sleeve is touching me now, but it's really not moving. If I move my arm, the shirt sleeve moves just a millimeter or two up or down. If I do little like rubbing, grooming movements, I'm moving rather slowly. Presumably, almost all of what touches our skin in life is slowly moving. So we took this likelihood function, the likelihood of every hypothesized tap 1 and tap 2 position, and we applied a low velocity expectation to our Bayesian computer model. And so this is nothing more than another bell-shaped curve, but it's plotting the prior probability the person might have about what's the velocity of movement on the skin. And it's speculating that zero velocity is probably the most common, that the most common thing that happens is something touching your skin doesn't move at all. But that it's possible that something could move up or down your skin with some speed, but very unlikely that it would move really fast, like 20 centimeters per second. That would be really unusual. If we just take this nice bell-shaped curve for the velocity expectation, the velocity prior, we can convert it into a prior in this diagram. If we know the time between the two taps was, for example, 0.2 seconds, and we know the velocity sigma, this is the standard deviation of this velocity expectation. So the smaller this is, the more the person expects things to just be very, very slow or stationary. We can change this to sort of a blob of expectation where the greatest probability a priori would be that the two taps are equal. So this is tap 1 equals tap 2 on this dotted line. But it's possible that tap 1 and tap 2 could be spaced apart. But if they occur really close together in time, they're not going to be too spaced apart because that would mean a really fast jump, right? So we can convert this velocity expectation to a prior in our space here, our pixels of x1 and x2 hypotheses. And then we just multiply every pixel here by the corresponding pixel here to do Bayesian inference. We take the product of the likelihood and the prior. And then this is our posterior. And you can see that the posterior is another blob of uncertainty, but it's like the likelihood function has been attracted towards the diagonal line because the prior was higher near the diagonal. So it's like we've taken the likelihood function and scooted it a little bit towards the line where the 2 points are closer together, okay? And what's shown here, this open circle here is the maximum likelihood estimate, and the closed one is the maximum a posteriori, the map estimate. So you can see that the map estimate has moved the 2 points closer together than they really were, which is actually the illusion that occurs. I should say there's really nothing new under the sun, and something very similar to our model had already been proposed earlier by some visual researchers who used it to explain a variety of visual motion illusions. And since our work, other researchers have also speculated in the auditory system about a low speed prior expectation. So I think this is probably a really general thing that's happening in perception. So just to illustrate what's happening here again, we have our likelihood function that has an uncertainty sigma sub s. If you think of these diagrams of time and space again, these are the real taps. So these are like the maximum likelihood estimates, but the brain is uncertain, and I'm showing little arrows going plus and minus 1 sigma s, indicating the brain's uncertainty about where it was tapped. And then we have a prior expectation that things move slowly, and that expectation has a sigma of sigma v for velocity. And as I mentioned, if you think something's going to move slowly, so a stationary movement is just a horizontal line here, no movement. And a slope going up would be going to the wrist, and going down would mean you have movement back to the elbow. And what this is saying is that it's possible that you could have movement, but it's unlikely you're going to have really steep trajectories, not very fast movement, because the prior says that we expect things to move slow. But you can see that no matter how, whatever your expectation is for speed, if you move for a greater time, you can move a greater distance. So if we have a second between the two taps, then this prior here is going to be wider. The two taps could be further apart. If they're moving slowly, they can still go a much further distance with more time. So it turns out that the width of this diagonal low speed prior here is equal to t times sigma v, the time times the standard deviation of our velocity prior. Well, anyway, then when we get the map estimate by multiplying each hypothesis's likelihood by its prior, we can plot the map estimate here. So here the MLEs are the open circles again, and the MAP, the perceived trajectory, is the filled circles. And what you can see in this particular example is that the distance between the two taps was actually 4 centimeters between the two open circles, but the perceived distance was only, L star was only 2 centimeters, so half of the real distance in this example. To look more quantitatively at how our model matches human perception, we did simulations where we can vary the distance between the two taps. So we change the true length L. And when we change the true length L, we're making the first tap closer to the elbow and the second tap closer to the wrist. So that changes the location of the likelihoods. The prior is the same. We didn't change the time at all. So the prior is the same. All of these are 0.15 seconds and between taps. But what you can see is that the posterior, as you change the position of the taps, the posterior does change. It gets pulled by the likelihood, but it's always retarded, held back by the prior. So the MLE is increasing as we increase the distance between the two taps, these open circles. But the MAP, the distance between the filled circles, is also increasing, but not as much. So L star always underestimates L, but L star actually ends up increasing linearly with L, as was observed with humans. What if we change the time? If we change the time between the two taps while leaving the distance between them the same, so in each of these columns, the stimuli were at the same 2 places on the skin, but we increase the time from 0.05 seconds to 0.35 seconds. As I mentioned before, you can see that the prior here gets broader. It's the same velocity expectation, the same low speed expectation. But when there are longer time elapses, the brain realizes things can move a longer distance. And then what turns out to happen is that the MAP, the filled circle here, ends up approaching the MLE, the open circle, as time is increased. You can see that because you can see that the prior, if it gets very wide here, doesn't exert much influence when you do the multiplication. You're multiplying each pixel here by almost the same value here. The priors become much more equal across different hypotheses. So you get less length contraction. So it turns out because this is all done with mathematical formulas, we can derive an actual formula that tells us in our model what the perceived distance between two taps should be, L star, as a function of the true distance, L, and the time between them, T. And this depends only on, L star depends only on L and T, and on the sigma S, which is the spatial uncertainty, and the sigma V, the low speed expectation of the prior. So you can see here the likelihood with sigma S and the prior with sigma V, together with the time and the length between the two taps, tells us exactly where the map estimate's going to be, the distance between these two taps. And so then we can apply that formula that we call the perceptual length contraction formula, and this is my last sort of technical slide. We can apply it to the data I showed you before, which were collected from other labs. And so I'm just going to show the fit of our model with solid curves here to those data. And I think you can see it fits rather well to previously published data. And we've since now done our own experiments to test it even more rigorously. And it seems to be holding up in general quite well. It may not describe every detail of human tactile perception. I'm sure it doesn't. But I think may have captured something here. We may have captured the rabbit, so to speak, and figured something out about how the illusion is working. So with that, I will stop. I know that was a lot of technical information. I hope, though, that I've convinced you that perception is an inherently uncertain process because different events in the world, like different baskets of apples, can produce equivalent sensory neural responses like the green apple. So taps to different regions of the skin can produce the same patterns of action potentials. And that's why we have that blob of uncertainty, the likelihood function in tactile perception, for example. And we can model perception as a Bayesian process. A Bayesian inference process combines likelihoods and priors to produce posteriors. What we've shown, it doesn't necessarily mean that the brain is doing that, but it's consistent with the idea that the brain is doing something very similar, if not Bayesian inference. If you want more information, I actually teach a course every winter semester, Psych 4 KK3 Bayesian inference. It's also, I have a grad students can sit in the same course, and I give them harder homework assignments and tests. And also I have postdocs who can sit in. So this year I actually have 2 postdocs who are just auditing the course. Of course, I encourage them to do the homework assignments for their own benefit. But I don't make the TAs mark their homework assignments because that wouldn't be fair. But I'm happy to have people sit in on the course. And then we also have some publications from the lab where we've described the model in more detail and a first empirical study where we tested the model. We have a second empirical study we've completed, but I haven't had any time to write it up. I feel really guilty about that, but I'm just COVID times. It's just too hard to do anything else. So, and so finally, I just like to acknowledge the students who have worked on this over the years, grad students as well as undergrads. You can see V and Akash prominently depicted, and my source of funding, which is NSERC. And I'd be happy to take any questions you might have. In fact, I think I'll unmute you all now. And you can just jump in with any questions.